Johnson Review Podcast. As always, I have provided a numbered list of sources used in the creation of this podcast. Today's topic is a rare disease by the name of short/branch-chain specific Acyl-CoA Dehydrogenase Deficiency. The abbreviated name for this disease, which I'll be using for the remainder of this podcast, is SBCAD deficiency, or SBCAD deficiency. SBCAD deficiency is an incredibly rare autosomal recessive genetic disease with fewer than 1,000 cases reported in the United States, and that statistic is taken from source number 8 on our source list. The symptoms of this disease are typically noticed within 23 months of childbirth, and the early symptoms include poor feeding, fatigue, irritability, and vomiting. Later, these symptoms can progress into difficulty breathing, seizures, neurological developmental delay, cognitive deficiency, and in extreme cases, patients can fall into a coma. These symptoms can be present on a wide range of severity, from extremely severe to extremely mild or even totally absent, according to sources 8 and 5. There are in fact a few reported cases of SBCAD deficiency where none of the symptoms that I just listed above are present. Now, at this point, you may be thinking to yourself, well, how is that even possible? How could we even know that an individual has this disease if they display none of the symptoms? In order to understand that, we'll have to dive deeper into the background and the mechanism of how SBCAD deficiency works. So, to provide a little bit of background for you all, I'd like to start by discussing the gene that this disease affects and its normal function. Then we'll get into the effect that SBCAD deficiency has on that gene and how that leads to the broader symptoms that we see from this disease. But before that, a word from today's sponsor, Keurig. Keurig has recently blasted its way into the coffee industry by revolutionizing quick and easy access to a delicious, crisp, and cognitively stimulating cup of coffee. I actually got this one for free here. From the dumpster outside my apartment, of course, not from Keurig, but still, it was free, and it works fantastically. Let me show you how it works. Just got to fill this puppy up right here with some cold water. Did that ahead of time. And then you press this power button right here. Boom. We're heating up. Now this is just going to take a little bit, and while that's heating up, let's get into this gene information. Short/branch-chain specific Acyl-CoA Dehydrogenase is the name of both the gene and the protein that the gene encodes. When referencing the gene, it is typically abbreviated as ACAD-SB or ACAD-SB. When referencing the protein, it is abbreviated as SBCAD. Another name for the protein is 2-methylbutyryl-CoA dehydrogenase, which I'll explain in just a minute why that is another name for the protein. So what does this protein do? Mind the sound. Trust me, it's worth it. So, the SBCAD protein is an enzyme involved in the metabolism of the amino acid L-isoleucine. When isoleucine is consumed, it goes through a series of metabolic reactions, one of which produces the intermediate 2-methylbutyryl-CoA. The SBCAD enzyme protein catalyzes the dehydrogenation of this intermediate, allowing it to continue along its metabolic pathway, eventually leading to the production of ATP. This information is from sources 2, 3, 4, and 5. Now, at this point, I'd like to provide you all with a little bit of general background on proteins and specifically protein structure. Proteins are considered polymers made up of amino acid monomers. due to the structure of a single amino acid containing, just as the name suggests, an amino group as well as an acid group. The overall structure of the protein itself contains an amino terminus, which is the exposed end of the exposed amino group end of it, which is synthesized first. Alternatively, it's referred to as the end term. And they also have a carboxylate terminus, which is the exposed carboxylic acid, of course, deprotonated at biological pH being around 7 pKa for a typical carboxylic acid is around 2.2. So then it is termed the carboxylate terminus, or the C term. These are synthesized at the end of proteins. Amino acids have a main chain as well as a side chain. The main chain is the same for all the amino acids. It contains the amino group and then a central carbon and then the carboxylate carbon. The side chain it varies between the amino acids. It's what determines the different kinds of amino acids, is they have a specific side chain which hangs off of that middle alpha carbon, the central carbon that I mentioned earlier. The specific amino acids that compose a protein are what's considered the protein's primary structure. A protein's secondary structure refers to the localized shapes that the amino acids in that region take on due to the non-covalent interactions between the amino acids' main chains. Some common types of secondary structure include alpha helices and beta sheets. The tertiary structure of a protein is the overall broad view structure of the different combinations and orientations of its secondary structure. The tertiary structure is mainly due to non-covalent interactions between the amino acid side chains rather than the main chains. And finally, if you didn't think there were enough degrees of structure for proteins, there is sometimes a quaternary structure, which only applies when multiple individual proteins associate into a protein complex, which, as I'll discuss later, the protein being discussed right now, Acyl-CoA Dehydrogenase, does in fact have a quaternary structure. And the last term that I would like to define related to protein structure is the term domain. A domain is a region of the protein which is typically responsible for a specific function. It is capable of folding into a functional form on its own, and it is common among several different proteins. It has a consistently identifiable pattern. The Acyl-CoA enzyme contains 3 major domains, with the first one being the N-terminus domain. This ranges from amino acids numbered 58 through 169, and it is made up of alpha helices. And you might be thinking, how can the N-terminus domain start at amino acid sequence number 58 if the N-terminus is the first amino acid? Well, there is something called post-translational modification. All amino acids start with a methionine and several other amino acids after that. And during post-translational modification, which is modification that occurs to a protein after it has been synthesized, typically cleaves off a certain beginning portion. And in this case, there is a specific signal sequence that is cleaved off, and we'll discuss that more later. The next is the middle domain, ranging from amino acid numbers 173 to 268, and it contains a beta barrel fold, which is kind of a more specific form of a secondary and tertiary structure blended together. It's essentially a lot of beta sheets, antiparallel beta sheets that come together to form a sort of barrel type structure, as the name would suggest. Finally, the C-term domain ranges from amino acids number 280 to 428, and it contains an all-alpha 4-helical up and down bundle. The active site is located on the C-term domain, with the catalytic residue being a glutamate at amino acid position 381. This information is taken from source number 3 on our source list. And would you look at that? The curie is now heated. So, now all I have to do at this point is open this puppy up, throw in my 30-cent off-brand K-cup. Don't tell them that I'm not using the actual Keurig cup, so they might pull my sponsorship, which doesn't actually exist anyway. But pop that sucker down, press this middle button here for my desired quantity of coffee. It's rather late, so I can't be doing that much. And you may find that the sound is a little bit unappetizing, to be honest with you. But trust me, it's good. We'll just give this a moment. You may think I'm crazy. I like my coffee black. Some people will say it's not the best for your teeth or for your stomach, and those people may be right. But I just like it black. What can I say? That's the sound of a good cup of coffee, trust me, people. That's good stuff right there. Turn this puppy off, otherwise it'll start heating up again automatically, which we don't necessarily need. And look at that. Crisp. Wonderful. Now back to business. As I mentioned, the enzyme is involved in ATP production. So therefore it is located within the mitochondria, specifically within the mitochondrial matrix, according to source number 2. Similarly to all other mitochondrial enzymes, SBCAD is fully translated by ribosomal complexes in the cytosol and remains unfolded. Chaperone proteins then bind to this mitochondrial signal sequence, which is the sequence that I was referring to earlier that gets cleaved off, but we'll get to that. So the mitochondrial signal sequence basically targets it to the mitochondrial matrix. So how it gets there is these chaperone proteins bind to the protein to ensure that it remains unfolded while it's being transported into the mitochondria. Upon reaching the outer skirts of the mitochondria, SBCAD passes through the translocase of the outer mitochondrial membrane, which is commonly referred to as the TOM complex, or the TOM complex, and then it passes through the translocase inner mitochondrial membrane, or the TIM complex. The mitochondrial signal sequence is then cleaved off by mitochondrial processing peptidase and folded in order to perform its proper function, according to source number 1. Now at this point we have covered the gene/protein's normal structure, specific function, as well as its subcellular location. So what is the basis for the deficiency of this enzyme in patients possessing this disorder? Well, a paper titled Isolated 2-methylbutyrylglycinuria caused by short/branch chain Acyl-CoA Dehydrogenase Deficiency, identification of a new enzyme defect, resolution of its molecular basis, and evidence for distinct Acyl-CoA dehydrogenases in isoleucine and valine metabolism. Yes, it's quite a mouthful of a title. Scientific journals are not exactly known for being succinct. Anyway, this paper concluded that it is actually the effect of a single missense point mutation, changing the guanine at the base pair position 1228 on the ACAD-SB gene to an adenine. Guanine at position 1228 is actually the terminal base pair of exon 10 on the ACAD-SB gene. So G1228, which is that guanine 1228 position, G1228 as well as the first two base pairs of exon 11 normally encode for a glycine. With the mutation, this codon produces a serine. However, the single point mutation does not simply result in a translated SPCAD protein with one incorrect amino acid. It is a much more deleterious mutation than that for two main reasons. Firstly, the mutation causes aberrant splicing of the exons during transcription of the ACAD-SP gene. This results in the total skippage of exon 10 in the transcription process, cutting out a large portion of the original gene. Secondly, because the terminal base pair of exon 10 is the first base pair of its codon, skipping it during transcription results in a frame shift for the remainder of the transcription process. And due to this frame shift, there is a premature termination codon present just 12 codons downstream of the frame shift according to source number 2. The authors of this paper discovered this mutation by sequencing and analyzing copy DNA of the ACAD-SP gene from someone with SPCAD deficiency. Copy DNA is DNA obtained through reverse transcription of a sample of mRNA from the patient. Upon observing this copy DNA, a 100 base pair deletion was discovered in exon 10. However, when the authors sequenced the SPCAD gene from the patient's genomic DNA, they did not observe that deletion, but they did find that guanine to adenine mutation that I was just discussing previously, meaning that the deletion present in the mRNA was not due to a 100 base pair deletion in the genomic DNA, but rather due to that aberrant splicing of the exons during the transcription of the ACAD-SP gene. So the variant protein that results from this mutation shows no detectable enzymatic activity and has trouble folding properly to form homotetramers, which is a term describing quaternary structure, meaning 4 of the exact same proteins combining together to form a complex, which is the typical behavior of the SPCAD protein. Notably, because the premature termination codon is close enough to the wild-type stop codon, the mutant mRNA is stable enough for translation to occur normally, meaning that the decreased SPCAD enzymatic activity in affected individuals is solely due to a deficiency in the enzyme's function rather than a deficiency in its quantity. So now that you all fully understand the effect of the genetic mutation, let's move on to how this deficiency of function in the SPCAD enzyme causes the broader scale symptoms that you actually can observe in patients with this disorder. In order to fully understand this, we must first cover the broader effects of the wild-type enzyme. So the wild-type enzyme, being in the mitochondria, has the most impact on cells rich in mitochondria, such as myocytes or muscle cells, and neurons, which I would assume you all know are nervous system cells. So mainly brain, stomach actually. A lot of people don't know there's a lot of neurons in the stomach. Either way, muscle cells, of course, use ATP to contract myosin II, inducing the shortening of the sarcomere, and as a result, motion. Neurons use ATP mainly for signal transduction in order to communicate with one another. A major way that neurons communicate is through the release of neurotransmitters and neuromodulators, such as dopamine, serotonin, or acetylcholine. These are released from the synaptic cleft of one neuron and transported into the surrounding neurons by dendrites. So quite directly, a dysfunctional SPCAD enzyme leads to an inability to metabolize L-isoleucine, therefore cutting off one major source of ATP from myocytes and neurons, leading to a physical and mental fatigue and dysfunctional state, according to source number 6. This also brings me back to my earlier posited question of how we can accurately diagnose someone with SP-CAD deficiency if they perhaps aren't displaying any symptoms. So due to the inability to metabolize isoleucine, individuals will have an extremely heightened levels of 2-methylbutyrylglycine in their urine, and this is termed 2-methylbutyrylglycinuria. So you can detect for that in patients' urine and identify whether or not they have this disorder, whether or not their symptoms are extreme or mild. Now, because these symptoms stem from the inability to metabolize isoleucine, as I've mentioned, patients would likely benefit from limiting their intake of isoleucine by following a low-protein diet. Now, that sounds good on paper, and indeed it probably can help, but you would also certainly question, what are the risks of engaging in a low-protein diet? What are the negative effects that may come from avoiding sources of isoleucine that also contain many other micronutrients that are extremely beneficial to the body, which is why a treatment option is certainly not exceptionally straightforward for somebody displaying extreme symptoms with this disorder. I suppose you could say in more extreme cases, the risk versus reward of limiting your protein intake is likely going to be more rewarding. You're likely going to benefit from limiting your protein intake if you have severe symptoms. Somebody with more mild symptoms, it's certainly something that is a very individual circumstance that you have to experiment on your own with. In addition to limiting the protein intake, you're going to want to get as many calories as possible, mainly from carbohydrates and fats, of course, which will aid in meeting the energy needs of neurons and myocytes and stave off some of those symptoms as well. A more advanced potential treatment option that has not yet been explored is direct enzymatic therapy, which would mean administering patients a functional form of the SP-CAD protein. There are many challenges associated with this type of treatment, such as the half-life duration of the administered enzyme, the potential immune response of the patient's body to the foreign enzyme, and developing a convenient method of administering the enzyme to patients so that they can do so frequently. So, although this is a challenging treatment option to begin thinking about developing, it has been shown to work in some diseases such as dyspepsia, which is chronic indigestion. Organizations have developed methods of administering the enzyme pepsin to aid with dyspepsia, according to source number 4. So I hope I've given you all a thorough understanding of this rare disease, SP-CAD deficiency, at this point. And I urge you all to go out and purchase yourself one of these bad boys as well. Thank you for watching.